Thank you very much for this opportunity for introducing my research. Let's go. In psoriasis, T-cell influx into the skin proceeds and induces neutrophil recruitment, which drives hyperkeratosis and plaque formation. In early skin lesions, neutrophil inflammasomes are activated, leading to netosis, loss production, and the release of cytokines and DAMPs, all of which exacerbate psoriasis. Neutrophil activation plays a key role in psoriasis pathogenesis. Upon rolling to arrest in microcirculation, neutrophil release DAMPs. One prominent DAMP is calprotectin. Once released, it primes neutrophils for enhanced inflammation during degranulation and inflammasome pore formation. Recently, we published a report on the dynamics of neutrophil recruitment and activation by comparing psoriasis patients with healthy controls. Employing an ex vivo flow channel, we demonstrated that psoriatic neutrophils roll slower and arrest more efficiently on E-Selectin and ICAM1 coated plate. We detected elevated plasma calprotectin levels, and this correlated with the extent of activated CD18 binding to ICAM1. The upregulation of activated CD18 supports more efficient neutrophil recruitment under shear flow. These previous studies led to our current hypothesis. This study is designed to gauge the neutrophil inflammasome activation and also to show that a key is mechanotransduction of selectin and integrin receptors. In vivo, neutrophils attach to vascular endothelium via E-Selectin and L-Selectin bonds under shear flow. Therefore, the bonds are subjected to mechanical stress. Since it is difficult to quantify neutrophil priming and inflammasome activation in skin tissue, we have assembled an in vitro flow cytometry-based assay. This mimics in vivo condition of shear flow and selectin bond formation using E-Selectin coated beads. This allows the neutrophil to feel a tension force on the bond. Neutrophils are isolated from fresh blood and shared in a test tube for 20 minutes. Then, activation is quantified in response to attachment to E-Selectin coated beads, with or without TNF-alpha and LPS stimulation. Cells are fixed and analyzed by flow cytometry. Flicker is a fluorescent dye that binds to caspase-1 of the inflammasomes, which can indicate the degree of NLRP3 inflammasome activity. Neutrophils are isolated from whole blood, stained with antibodies, and immediately fixed for this measurement. We observed that psoriasis neutrophils are more activated than healthy controls across all four of our activation markers. This demonstrates that psoriatic neutrophils already exhibit a higher activated state while in circulation. While it is not well known that stimulants like LPS and TNF-alpha activate neutrophils, we found that E-Selectin coated beads also effectively drive activation. Interestingly, while stimulation with LPS or TNF-alpha induces the shedding of CD62L, known as L-Selectin, stimulation with EB does not induce this shedding. Furthermore, EB demonstrated a synergistic effect when combined with LPS or TNF-alpha. This suggests that The stimulation triggered by E-Selectin-L-Selectin binding utilizes a distinct signaling pathway from that of LPS or TNF-alpha. The important point is that addition of both E-Selectin and DAMP or cytokine enables testing of conditions known to exist in psoriatic skin. Our principal component analysis revealed a specific subset of psoriasis patients who are high responders to E-Selectin stimulation. When looking at Flickr and high-affinity CD18, we found that in this high responder group, individual neutrophils do not show higher levels of activation per cell than healthy controls. Instead, they exhibit an increased overall percentage of activated cells within the total neutrophil population. In conclusion, we have established a method using E-Selectin-coated beads to mimic in vivo neutrophil activation and measure this process. We found that many psoriasis patients exhibit a higher baseline activation state compared to healthy controls. Furthermore, the E-Selectin pathway and integrin pathways are distinct but engage in synergistic signaling. Finally, a specific subset of psoriasis patients is highly sensitive to E-Selectin stimulation. Thank you for your attention.
Thanks a lot for this very nice talk. Any questions? Maybe I have one. Did you link it to any, if it's clinically or histologically a bit different? So there's like more neutrophils activated in the epidermis or these two subgroups. Does this correlate to anything in the skin? No, the two subsets you had, the one responded to the selectin or not. Is this and linked or correlated with a different clinical presentation, histological presentation, more neutrophils in the epidermis or different signature?
This small project, so all psoriasis patients are middle severity, like BSA around 10%. So now clinical manifestation is not different, our research.
Okay, thanks a lot.
Thank you.
Thank you.